General and special senses. Sensory information arriving at the central nervous system is transmitted up the spinal cord and into the brain. This carries information on 1. The location where the sensory information was detected, and 2. The nature of the stimulus that was detected. Each pathway carries information for a specific sensation, such as touch, pressure, or vision, from receptors in a specific part of the body. All other characteristics of the stimulus are determined by the pattern of the action potentials in the afferent fibers. This sensory coding provides information about the strength, duration, variation, and movement of the stimulus. The area monitored by a single receptor is called the receptive field. The larger the receptive field, the harder it is to localize the stimulus. For example, a touch receptor on your back may have a receptive field of 7 centimeters in diameter, while a receptor on your tongue may have a receptive field of less than a millimeter in diameter. As a result, we can describe a light touch on the back only generally, but we can be very precise about the location of a stimulus placed on the tongue. There are two categories of receptors: tonic receptors and phasic receptors. Tonic receptors are sensory neurons that are always active. Phasic receptors are normally inactive, but they become active for a short time whenever there is a change in conditions that they are monitoring. Phasic receptors provide information on the intensity and rate of change of a stimulus. Adaptation is a reduction in sensitivity in the presence of a constant stimulus. There are both peripheral adaptations and central adaptations. Peripheral or sensory adaptation is when a receptor or sensory neuron alters its level of activity. The receptor responds strongly at first, but then the activity along the afferent fiber gradually declines because of synaptic fatigue. Phasic receptors are fast to adapt, whereas tonic receptors are slow to adapt. Central adaptation occurs at the subconscious level. Central adaptation restricts the amount of information arriving at the cerebral cortex. The sensory information arriving at the central nervous system is called a sensation. A perception is a conscious awareness of a sensation. General senses refer to sensations of temperature, pain, touch, pressure, vibration, and proprioception or body position. General sensory receptors are distributed throughout the body. The special senses are smell, taste, equilibrium, hearing, and vision. The specialized receptors providing these sensations are structurally more complex than those of the general senses. These receptors are localized within complex sensory organs, such as the eye or ear. General sensory receptors are divided into three classes: exteroceptors, proprioceptors, and interoceptors. Exteroceptors provide information about the external environment. Proprioceptors monitor body position, and interoceptors monitor the conditions inside the body. A more detailed classification system using the nature of the stimulus detected divides general sensory receptors into four classes: nociceptors, thermoreceptors, mechanoreceptors, and chemoreceptors. Nociceptors respond to a variety of stimuli associated with tissue damage. Activation of these receptors causes the sensation of pain. Thermoreceptors respond to changes in temperature. Mechanoreceptors are stimulated or inhibited by physical distortion, contact, or pressure on plasma membranes. Chemoreceptors monitor the chemical composition of body fluids and respond to the presence of specific molecules. Thermoreceptors detect changes in temperature. There are two classes of thermoreceptors: cold thermoreceptors and warm thermoreceptors. These are found in the dermis of the skin, skeletal muscles, the liver, and the hypothalamus. Thermoreceptors are phasic receptors. They are very active when the temperature is changing. However, They quickly adapt when temperature becomes stabilized. For example, when you enter an air-conditioned classroom on a hot summer day, the temperature change seems extreme at first, but you quickly become comfortable as adaptation occurs. Mechanoreceptors are sensitive to stimuli that stretch, compress, twist, or distort their cell membranes. There are three classes of mechanoreceptors: tactile receptors, baroreceptors, and proprioceptors. Tactile receptors detect sensations of touch, pressure, and vibration. Baroreceptors detect pressure changes in the walls of blood vessels and in portions of the digestive, reproductive, and urinary tracts. Proprioceptors detect the positions of joints and skeletal muscles and are the most complex of the general sensory receptors. Fine touch is sensed by free nerve endings, tactile discs, and the root hair plexuses. Free nerve endings and tactile discs respond to light contact with the skin, whereas the root hair plexus responds to the initial contact with the hair shaft. Pressure and vibration are sensed by tactile corpuscles and lamellar corpuscles. Tactile corpuscles respond to initial contact and low-frequency vibrations. Lamellar corpuscles respond to initial deep contact and high-frequency vibrations. Deep pressure is sensed by bulbous corpuscles. The bulbous corpuscles respond to stretching and distortion of the dermis. Proprioceptors monitor the position of joints, the tension in tendons and ligaments, and the extension of muscular contraction. Muscle spindles are proprioceptors that monitor the length of skeletal muscles. Golgi tendon organs monitor the tension in tendons during muscle contraction. Chemoreceptors are specialized neurons that detect changes in the concentration of specific chemicals or compounds. For example, chemoreceptors in and near the respiratory centers of the medulla oblongata are sensitive to changes in pH and carbon dioxide levels. Chemoreceptors of the carotid bodies and aortic bodies are sensitive to changes in pH, concentrations of carbon dioxide, and concentrations of oxygen in the blood. The olfactory organs gives us our special sense of olfaction or smell. Olfactory reception occurs on the surface of olfactory cilia. When the odorous substance binds to its receptor on the cilium, the receptor membrane depolarizes. This depolarization triggers an action potential in the axon of the olfactory receptor. The olfactory pathway is only a two-neuron pathway. In fact, the olfactory sensations are the only sensations that travel directly to the cerebral cortex without synapsing at the thalamus first. Gustation, or taste, provides information about the foods and liquids we eat or drink. Gustatory epithelial cells, or taste receptors, are found inside of taste buds. These taste buds are distributed all over the tongue's surface and adjacent portions of the pharynx and larynx. These cells are stimulated by dissolved food molecules. Stimulation leads to action potentials that are sent to the gustatory cortex for interpretation. Cranial nerves 7, 9, and 10 carry the gustatory information to the gustatory cortex of the cerebrum. The ear is associated with the detection of two special senses: equilibrium and hearing. The ear is divided into three anatomical regions: the external ear, the middle ear, and the internal ear. The external ear is the visible portion, and it collects and directs sound waves to the eardrum. The middle ear is a chamber located within the petrous portion of the temporal bone. Structures within the middle ear amplify sound waves and transmit them to the appropriate portion of the inner ear. The internal ear contains the sensory organs for hearing and equilibrium. The external ear includes the outer fleshy auricle, which surrounds the passageway called the external acoustic meatus. The auricle protects the opening passageway and provides directional sensitivity to the ear by directing sound inward toward the tympanic membrane or eardrum. The tympanic membrane is a thin, semi-transparent connective tissue sheet, separating the external ear from the middle ear. The tympanic membrane is very delicate. The auricle and narrow external acoustic meatus protect the tympanic membrane from injury. The middle ear consists of the tympanic cavity, which consists of the auditory ossicles. The middle ear connects the tympanic membrane with the receptor complex of the internal ear. Also located in the middle ear is the auditory or eustachian tube. The auditory tube helps equalize the pressure in the middle ear with the external atmospheric pressure. The three auditory ossicles are the malleus, incus, and stapes. These bones are the smallest bones of the body. They connect the tympanic membrane with the receptor complex of the internal ear. These bones act as levers, transferring and amplifying sound vibrations from the tympanic membrane to the fluid-filled chamber within the internal ear. The sensory receptors for equilibrium and hearing are located within the internal ear. The receptors are found within the membranous labyrinth, which is filled with a fluid called endolymph. The bony labyrinth surrounds and protects the membranous labyrinth within a liquid called perilymph. The bony labyrinth is subdivided into three areas: the vestibule, the semicircular canals, and the cochlea. The vestibule and the semicircular canals form the vestibular complex. The cavity within the vestibule contains the utricle and the saccule. Hair cells within the utricle and saccule provide position and linear movement sensations. Hair cells within the semicircular canals are stimulated by the rotation of the head. The vestibular complex is part of the internal ear and provides equilibrium sensations by detecting rotation, gravity, and acceleration. It consists of the semicircular canals, the utricle, and the saccule. The anterior, posterior, and lateral semicircular canals are continuous with the vestibule. Each semicircular canal surrounds a semicircular duct. The anterior, posterior, and lateral semicircular ducts are continuous with the utricle. Each duct contains an ampulla that contains hair cells in a region called the ampullary crest. Each hair cell in the vestibule has 80 to 100 stereocilia and a single kinocilium. When external forces pushes against these processes, the distortion of the plasma membrane alters the rate in which the hair cells releases neurotransmitters. The receptors within each semicircular duct respond to one of these rotational movements. A rotation in the anterior-posterior plane, such as nodding yes, stimulates the hair cells of the anterior duct. A rotation in the horizontal plane, such as shaking your head no, stimulates the hair cells of the lateral semicircular duct. A rotation in the coronal plane, such as tilting your head from side to side, stimulates the hair cells of the posterior duct. The hair cells of the utricle and saccule are clustered in the oval maculae. The macula of the utricle is sensitive to changes in horizontal movement, whereas the macula of the saccule is sensitive to changes in vertical movement. Sensory fibers from the vestibular ganglia form the vestibular nerve. The vestibular nerve synapses on neurons within the vestibular nuclei, located at the boundary between the pons and the medulla oblongata. The two vestibular nuclei integrate and relay sensory information, as send commands to the other motor nuclei in the brainstem and spinal cord. The cochlear duct is located between two chambers filled with perilymph. The outer walls of the perilymph-filled chambers consist of dense bone, except at two small areas near the base of the cochlear spiral. These are the round window and the oval window. The round window is the more inferior of the two openings. It is a thin, membranous partition that separates the perilymph of the cochlear chambers from the air-filled chamber of the middle ear. The oval window is the more superior of the two openings. It is located within the cochlear wall, and the base of the stapes almost completely fills this oval window. The sensory receptors of the internal ear are called hair cells. Hair cells are highly specialized mechanoreceptors. They are sensitive to the movement of their stereocilia. The hair cells of the cochlear duct are located within the spiral organ or organ of Corti, located in the cochlea. This sensory structure rests on the basilar membrane, which separates the cochlear duct from the scala tympani. The hair cells of the spiral organ are arranged in two longitudinal rows, an inner row and an outer row. These hair cells contain stereocilia that make contact with the overlying tectoral membrane. The tectoral membrane is firmly attached to the inner wall of the cochlear duct. When a portion of the basilar membrane vibrates up and down, the stereocilia of the hair cells become bent. The frequency or pitch of a sound is determined by what part of the basilar membrane moves the most. Movement of the stapes sets up pressure waves within the perilymph. These waves distort the cochlear duct and the spiral organ, stimulating the hair cells. High frequency or high pitch sounds affect the basilar membrane near the oval window, whereas the lower frequency sounds affect the basilar membrane in areas that are further away from the oval window. The intensity or volume of the sound is determined by the amount of distortion in the basilar membrane. This is dependent on the amount of force applied to the oval window. This relationship provides a mechanism for detecting the intensity of a sound. Here are the steps in the production of auditory sensation. 1. Sound waves arrive at the tympanic membrane. 2. Movement of the tympanic membrane causes displacement of the auditory ossicles. 3. Movement of the stapes at the oval window establishes pressure waves in the perilymph of the scala vestibuli. 4. The pressure waves distort the basilar membrane on their way to the round window of the scala tympani. 5. Vibration of the basilar membrane causes hair cells to vibrate against the tectoral membrane, resulting in hair cell stimulation and neurotransmitter release. And 6. Information concerning frequency and intensity of stimulation is relayed to the central nervous system over the cochlea nerve. The auditory pathway is a 4-neuron pathway ending at the auditory cortex of the cerebrum. A large number of collateral fibers terminate in motor nuclei in the brainstem and initiate auditory reflexes of varying kinds. Humans rely on vision more than any other special sense. As a result, the visual cortex is several times larger than the cortical area of any other special sense. The eyelids act like windshield wipers. Continual blinking keeps the surface lubricated and free from dust and debris. They also protect the delicate surfaces of the eye by closing firmly. The eyelashes along the margins of the eyelids are very strong hairs. Sensory structures in the root hair plexus monitor each eyelash. Movement of the hair triggers the blinking reflex, which prevents foreign matter from reaching the surface of the eye. The conjunctiva is the layer of epithelium covering the inner surface of the eyelids and the outer surface of the eye. A constant flow of tears keeps the conjunctival surface moist and clean. Tears reduce friction, remove debris, prevent bacterial infection, and provide nutrients and oxygen to the epithelium. The lacrimal apparatus produces, distributes, and removes tears. The lacrimal apparatus of each eye consists of 4 things: a lacrimal gland, the superior and inferior canaliculi, a lacrimal sac, and a nasolacrimal duct. The wall of the eye consists of 3 layers: the outer fibrous layer, the intermediate vascular layer, and the inner layer, or the retina. The eyeball is hollow, and its interior is divided into 2 cavities. The large posterior cavity, also known as the vitreous chamber, contains the gelatinous vitreous body. The smaller anterior cavity has two chambers: the anterior chamber, located between the cornea and the iris, and the posterior chamber, between the iris and the lens. The shape of the eye is established by the vitreous body and the clear aqueous humor filling the anterior cavity. The fibrous layer is the outermost layer of the eye. It consists of the sclera and the transparent cornea. The sclera is the white surface of the eye and covers most of the ocular surface. The cornea is the part of the fibrous layer that is transparent and is continuous with the sclera. The iris is seen through the transparent surface of the cornea. The iris contains blood vessels, pigment cells, and two layers of smooth muscles that are part of the intrinsic eye muscles. Contraction of the intrinsic eye muscles changes the diameter of the pupil, which is the central opening of the iris. The diameter of the pupil decreases when sphincter pupillae contract. Conversely, the diameter of the pupil increases when the dilator pupillae contract. The iris is attached to a ciliary body. The ciliary body consists of ciliary muscles, which are a muscular ring projecting interiorly into the eye. The ciliary zonule or suspensory ligament is the ring of fibers that attaches the lens to the ciliary processes. This adjusts the shape of the lens to focus the image onto the retina. The choroid is a vascular layer within an extensive capillary network that delivers oxygen and nutrients to the retina. The inner layer, or retina, consists of two distinct layers: an outer thin layer called the pigmented layer, and a thicker inner neural layer, which contains the visual receptors and associated neurons. The outermost layer contains the visual receptors. There are two types of photoreceptors: rods and cones. Rods do not detect different colors of light. However, they are very light sensitive and enable us to see in dimly lit rooms at twilight or in pale moonlight. Cones detect different colors and therefore provide us with our color vision. There are three types of cones: red, green, and blue, and their stimulation in various combination provides the detection of the different colors we experience. As you move toward the center of the retina, the density of rods gradually decreases, and the density of cones increases. This area is known as the macula, and it is the area in which the visual image arrives after passing through the cornea and the lens. The macula has no rods. The highest concentration of cones occurs at the center point of the macula. This area is called the fovea centralis, or simply the fovea. The rods and cones synapse with neurons called bipolar cells. Horizontal cells at the same level form a network that inhibits or promotes communication between different photoreceptor and bipolar cells. Bipolar cells then synapse with the layer of ganglionic cells. Amacrine cells, also found in this level, modulate the communication between the bipolar cells and the ganglion cells. The ganglion cells are the first-order neurons in the optic pathway and are the only cells in the retina generating action potentials that will travel to the brain through the optic pathway. The optic disc lies just medial to the fovea centralis. The optic disc is the origin of the optic nerve or cranial nerve 2. From this point, the axons turn and penetrate the wall of the eye, proceeding to the brain. There are no photoreceptors or other retinal structures at the optic disc. Because the light striking this area goes unnoticed, it creates what is commonly called the blind spot. You do not notice a blank spot in your vision field because the involuntary eye movements keep the visual image moving, allowing the brain to fill in the missing information. The chambers of the eye include the anterior, posterior, and vitreous chambers. The anterior and posterior chambers are filled with aqueous humor. The circulation of the aqueous humor forms a fluid cushion that provides an important route for transporting nutrients and wastes. The vitreous chamber contains the vitreous body or vitreous humor. This gelatinous mass maintains the shape of the eye, supports the posterior surface of the lens, and supports the retina by pressing the neural layer against the pigmented layer. The lens focuses the visual shape on the retinal photoreceptors by changing its shape. A dense fibrous capsule covers the entire lens. The capsular fibers intermingle with those of the ciliary zonule. As tension in the ciliary zonule increases, the lens flattens. With the lens flattened, the eyeball is able to focus on distant objects. When the ciliary muscles contract, the ciliary body moves towards the lens. This movement reduces the tension in the ciliary zonule, and the lens assumes a more spherical shape. In this spherical position, the eye is able to focus on nearby objects. The visual pathway is a 3-neuron pathway, beginning in the ganglion cells of the retina and ending in the visual cortex of the cerebrum. Each rod and cone monitors a specific receptive field. A visual image results from processing information provided by the entire receptor population. A significant amount of processing occurs in the retina before the information is sent to the brain because of the interactions between various cell types. The right and left optic nerves reach the diencephalon after a partial decussation at the optic chiasm. At the optic chiasm, approximately half of the fibers proceed toward the lateral geniculate nucleus of the same side of the brain, while the other half cross over to the lateral geniculate nucleus of the contralateral side. Visual information from the left half of each retina synapses at the lateral geniculate nucleus of the left side. Likewise, visual information from the right half of each retina synapses at the lateral geniculate nucleus of the right side. The lateral geniculate nuclei act as a switching center, relaying visual information to reflex centers in the brainstem and cerebral cortex. Some visual information, however, bypasses the lateral geniculate nuclei altogether. For instance, the reflexes controlling eye movement are triggered by information that bypasses the lateral geniculate nuclei to synapse in the superior colliculi. The perception of vision results from the integration of information arriving at the visual cortex of the occipital lobes of the cerebral hemispheres. The visual cortex contains a sensory map of the entire field of vision. Also, each eye receives a slightly different image. The association and integrative areas of the cortex compare the two images and use them to provide us with depth perception. The partial crossover occurring at the optic chiasm ensures that the visual cortex receives a composite picture of the entire visual field. Many centers in the brainstem receive visual information from the lateral geniculate nuclei or collaterals arising from the optic tracts. Collaterals bypassing the lateral geniculate nuclei synapse in either the superior colliculus or the hypothalamus. The superior colliculus of the mesencephalon issues motor commands, controlling subconscious eye, head, and neck movements in response to visual stimuli. Visual inputs to the suprachiasmatic nucleus of the hypothalamus and the endocrine cells of the pineal gland establish a daily pattern of visceral activity that is tied to the day-night cycle. This is called the circadian rhythm, and it affects metabolic rates, endocrine function, blood pressure, digestive activities, the sleep-wake cycle, and other physiological processes. Thank you for watching.